Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Okay, so imagine this tiny, invisible intruder. A virus, right? And it's living inside maybe half the people listening right now. Often, totally silent, no symptoms.
Right. Most people don't even know they have it.
But then for others, for the really vulnerable people, this same virus, cytomegalovirus, CMV, it can become, well, deadly.
Absolutely. It's a huge threat in certain situations.
So today, that's what we're diving into. There's some really exciting new research, a whole new way potentially to fight CMV.
And it's critical research because, as you said, while many people carry CMV without issue, for immunocompromised individuals, think organ transplant recipients, people with AIDS, it's incredibly dangerous.
Life-threatening complications.
Exactly. And there's another really tragic aspect, congenital CMV. If a mother passes it to her baby during pregnancy,
Oh, right.
it can cause permanent hearing loss. It's actually a leading non-genetic cause of deafness in newborns.
That's heartbreaking. And it really highlights why we need better options, doesn't it? Because the current tools, well, they have problems.
They do. The antiviral drugs we have now, first, they're expensive.
Yeah.
And they come with some pretty serious side effects. We're talking potential bone marrow suppression, kidney damage. It's not trivial.
And the virus itself is getting smarter, isn't it? Like resistance is becoming a real issue.
It is, unfortunately. We're seeing more strains that just don't respond well to the existing drugs.
So drugs are tricky. What about harnessing the immune system directly? Things like T-cell therapies.
Yeah, things like adoptive T-cell transfer or CAR-RT therapy, they can be effective, definitely. But there's a major bottleneck.
Speed,
exactly.
Mm-hmm.
Preparing these therapies is complex. It involves taking a patient's cells, modifying or expanding them outside the body. It takes time.
How much time are we talking?
It could be anywhere from, say, 28 to 44 days, nearly a month, sometimes more.
Wow. And if you have a severe, fast-moving infection?
That's simply too long. You don't have that kind of time.
Okay, so the current options are maybe too harsh, or the virus outsmarts them, or they're just way too slow for urgent cases.
That sums it up pretty well.
So that's our mission for this deep dive. We need to unpack how scientists are trying to engineer a smarter, faster way to use our own immune system against CMV.
Using the body's own defenses, but giving them a major boost and clear directions.
Right. We're looking at some fascinating findings, mainly from a study in Science Advances, but also pulling in some health news context. How can we basically give our immune system a really precise, rapid command to go find and destroy these CMV-infected cells?
And this is where it gets really interesting, technically speaking.
Yeah, tell us about it, this new approach from UCLA researchers.
Yeah.
They're using something called T-cell redirecting bispecific antibodies.
TRBAs, that's right, TRBAs. Think of them as like molecular matchmakers, or maybe a tiny engineered bridge.
A bridge. Okay, explain that.
So they're antibodies, but they're bispecific. That means they're designed to grab onto two different things at the same time.
Two things. What are they connecting?
Okay, so one end of this bridge is engineered to bind very specifically to a marker called CD3 epsilon.
CD3. That's on our T-cells, right? Our killer T-cells.
Exactly. CD3 is part of the complex that T-cells use to get activated.
Yeah.
So binding to CD3 is like pushing the go button on the killer T-cell, the CD8 plus T-cell specifically.
Got it. Killer T-cell activation. What's the other end of the bridge grabbing?
The other end is designed to bind to a protein on the virus-infected cell, specifically a protein called glycoprotein H, or GH.
GH. And that's on the surface of cells infected with CMV?
Yes, and importantly, GH is pretty essential for the virus. It helps CMV get into new cells, so it's a stable target, and it's right there on the outside of the infected cell.
Okay, so the TRBA connects the killer T-cell directly to the CMV-infected cell, like handcuffing them together.
Kind of, yeah. This cross-linking, as it's called, physically pulls that killer T-cell right up close to its target.
And just being close is enough?
Well, the binding to CD3 and the proximity triggers the T-cell. It activates it. The T-cell then does what it's supposed to do. It clusters around that infected cell and initiates the killing process, destroys it directly.
And does it do anything else, like signal other immune cells?
Yes, that's another key part.
Yeah.
This activation also causes the T-cell to release important immune signals, cytokines, like interferon gamma.
Interferon gamma, okay. And that helps how?
Interferon gamma is a powerful antiviral signal. It ramps up the overall immune response, inhibits the virus from replicating, and makes other immune cells more effective. It basically sounds the alarm and calls in reinforcements.
Wow. It really does sound like a guided missile system for the immune system, but building something like that, a molecule that does two specific jobs, that must be complex. How did they actually design these TRBAs?
It's definitely sophisticated molecular engineering. The UCLA team wanted to tackle those limitations. We talked about the speed, the side effects, the resistance.
Right.
So their goal was to make something that could quickly get the immune system engaged to clear out those CMV cells. They essentially started with parts of known antibodies,
Okay.
one part that binds to the T-cells CD3, and another part that binds to the CMV's GH protein.
And just stuck them together, or was it more involved?
Oh, much more involved. They used a couple of really clever antibody engineering strategies to make sure the final molecule worked well.
Like what?
Well, one approach was called the crossover dual variable design, or CODV. It sounds complicated, but the idea is to arrange the binding parts on the antibody chains in a specific sort of reversed order.
Why do that?
It helps prevent the different parts of the antibody molecule from physically getting in each other's way, what scientists call steric hindrance. It makes the whole thing more stable and better able to bind both targets efficiently.
Okay, so better geometry, basically. What was the other strategy?
The other was maybe a bit more straightforward conceptually. They took a single chain version of the part that binds CMV. And basically tacked it onto the end of the heavy chain of the antibody part that binds CD3, a different way to link the two functions.
Okay, so they built these potential molecular bridges. First step must have been checking if they actually, connected to the right things.
Exactly, basic validation. They needed to confirm that these engineered antibodies could actually bind and bind well to both the CD8 plus T-cells and to cells infected with CMV.
And did they?
They did. The binding tests showed high affinity, meaning they latched on strongly and specifically.
Okay, so the bridges connect. Step 2, do they actually work? Can they get the T cells to kill the infected cells?
Right, the functional test. This is where they did co-culture experiments. They mixed human CD8 plus T cells together in a dish with human cells that were infected with CMV.
How could they tell which cells were infected?
Ah, clever trick. They engineered the infected cells to produce a green fluorescent protein, GFP. So the infected cells literally glowed green under a microscope.
Nice. Easy to track.
Very. Then they added their different TRBA constructs to this mix.
And what happened?
The results were pretty dramatic. They saw a significant drop in the number and even the size of those green, glowing infected cells.
So the TRBAs were helping the T cells clear the infection.
Yes. And importantly, it was dose dependent, meaning the more TRBA they added, the better the clearance. That strongly suggests the effect was directly due to the engineered antibodies.
Okay, that's compelling. But did they confirm the T cells were actually getting activated, releasing those signals like interferon gamma?
They did. They measured the cytokine levels in the culture medium. And just as expected, when the TRBAs were present, the levels of interferon gamma shot up.
So confirmation of T cell activation.
Right. It wasn't just that the infected cells were vanishing. They could show the T cells were being switched on by the TRBAs, releasing these potent antiviral signals. It really ties the mechanism together.
And they even visualized it, didn't they, with microscopy?
Yes. This is a really cool part, I think.
Yeah.
They used fluorescence microscopy to literally watch it happen.
What did they see?
They tagged the T cells so they glowed red. The infected cells, remember, glowed green.
Right, red T cells, green infected cells.
And when they added the TRBAs, you could actually see the red T cells swarming around, clustering really tightly against the green infected cells.
Wow.
And then over time, you could observe those green cells starting to break apart to disintegrate, right where the red T cells were attached.
So you could see the bridge working, the T cells being recruited, engaging, and killing.
Exactly. There's a very clear visual confirmation of the whole concept. Pretty ingenious stuff.
It really is. Okay, so the science in the lab looks fantastic, but let's bring it back to the bigger picture. What makes these TRBAs potentially better than maybe earlier attempts or the existing options we discussed? What's the real-world advantage here?
Well, there are several key advantages that make this approach really stand out. First and foremost is speed and efficiency.
Compared to the cell therapies.
Right. Unlike adoptive transfer or CAR-T, which need that long preparation time, weeks, remember, these TRBAs are off the shelf, ready to go.
Meaning you could theoretically just infuse them into a patient.
Potentially, yes. They act immediately. No need for taking cells out, modifying them, growing them. That rapid response could be absolutely crucial for someone with an acute, dangerous infection.
That makes sense. Time is critical. What else?
Another major advantage is how long they last in the body. persistence.
Right. Some earlier antibody drugs didn't last long.
Exactly. Some of the first-generation bispecifics, like molecules called BiTEs, had very short half-lives, maybe only a couple hours.
Which means you'd need continuous infusion, right? Hooked up to an IV all the time.
Pretty much, which is challenging for patients. But these new TRBAs were designed differently. They include a part of the antibody called the IgG-Fc domain.
The FSC domain. What's it do?
It allows the antibody to interact with a natural recycling mechanism in our bodies involving something called the neonatal Fc receptor, or FcRn.
Okay.
Basically, this interaction protects the antibody from being broken down too quickly. It extends its life.
So how long do these TRBAs last?
The FEC domain gives them a much longer half-life, typically in the range of 10 to 14 days.
Days versus hours. That's a huge difference.
Massive. It opens up the possibility of intermittent dosing, maybe an infusion every week or two instead of continuous infusion.
Yeah.
Much more practical and patient-friendly.
Definitely. Any other advantages to the design?
Well, these TRBAs also have 2 binding sites for CMV and 2 for the T-cells on each molecule.
2 arms for each target.
Sort of, yeah.
Yeah.
Now, comparing potency directly with older designs is tricky because labs use different tests.
Sure.
But having multiple binding sites could theoretically increase the overall binding strength and potency, make it grip tighter, essentially.
And the target itself, GH. You mentioned that was important.
Very important. Targeting GH is a good strategy because it's highly conserved.
Meaning it doesn't change much between different CMV strains?
Exactly. And it's vital for the virus to infect cells. So it's harder for the virus to just mutate GH to escape the antibody without also hurting its own ability to spread. Makes it a robust target.
Okay, so faster, longer lasting, potentially more potent, good target. The implications for patients seem really significant.
They really are. Dr. Otto Yang, who led the study, put it plainly. He said, this could be a potentially new way to harness the immune system against this virus.
And he mentioned specific groups.
Yes. He specifically pointed to transplant patients, AIDS patients with severe infections, and also children at risk of deafness from CMV, people who desperately need better options.
It really does sound like a genuine alternative to those tough antivirals with the side effects and the resistance problems.
But it offers hope for that, yes.
Yeah.
If this pans out, it could shift how we manage these persistent, difficult viral infections.
It feels like a move towards more precision medicine, right? Using the body's own system, but guiding it very specifically.
That's a great way to put it. Instead of just carpet bombing with antivirals, you're providing this highly targeted immune boost right where it's needed.
Of course, we have to be realistic. You mentioned comparing potency is hard. Are there other limitations or next steps we should be aware of?
Absolutely. While the lab data is exciting, these are still preclinical results. The big hurdle, as Dr. Yang himself pointed out, is moving this forward.
Meaning?
It needs commercial interest, funding, and then the crucial step, testing in human clinical trials.
Right. To see if it's safe and effective in actual patients.
Precisely. That's the only way to know for sure if this promise translates into a real therapy.
So summing this all up then, what's the key takeaway for someone listening?
I think the core idea is this clever engineering. Creating these bispecific antibodies that act as bridges, rapidly connecting our killer T-cells to CMV-infected cells.
And triggering a targeted destruction.
Exactly. Offering a potentially faster, longer-lasting, and maybe safer way to combat CMV, especially for those most vulnerable.
It really shows the power of understanding the immune system at a molecular level, doesn't it? Designing tools to enhance its natural function.
It absolutely does. We're learning how to speak the immune system's language in a way and give it very specific instructions.
So looking ahead, this deep dive suggests the future of fighting viruses like CMV might involve less reliance on traditional drugs and more on these kinds of smart immune-boosting therapies.
It certainly points in that direction. A future where we can deploy our own defenses with incredible precision and speed.
It makes you wonder, if this works for CMV, what other chronic or stubborn infections could we target this way? Herpes viruses, HIV maybe, other persistent pathogens.
That's the exciting long-term question, isn't it? The potential applications could be quite broad if the platform proves successful.
A fascinating glimpse into how science is constantly finding new ways to empower our own bodies. Really impressive work.
Thanks for tuning in to the Health Headliner podcast by To-Do Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.